Now we're going to transition to talking about regulation of all of these things. So the first thing that I really want to highlight here is beginning with the effect of sympathetic stimulation on the kidney. Big picture wise, the renin, angiotensin, aldosterone, and sympathetic nervous systems want to accomplish a big goal, and that big goal is to retain water. And they retain water by retaining sodium and then kicking out potassium. And that is the kind of big picture goal of sympathetic nervous system stimulation. Now, systemic sympathetic nervous system stimulation can then go on and activate the local endogenous renal sympathetic nerve endings. And when that happens, three big effects occur. First, there is a vasoconstriction of the renal arterioles, and I'll go into more detail on that in a few more slides, the net effect of which results in a decrease in glomerular filtration rate. Second, it increases tubular reabsorption of water and salt. And then third, systemic sympathetic stimulation via local renal sympathetic nerves activates the renin-angiotensin-aldosterone system, which further increases tubular reabsorption of sodium. Now this is the same sort of set of information. Sympathetic stimulation results in three big effects, and those same three effects are listed out here in more of a graphic form rather than written out as on the previous slide. The first one is via activation of alpha-1 adenoreceptors on arterial resistance vessels, aka afferent and efferent arterioles, you result in a powerful vasoconstriction. Now, the alpha-1 adenoreceptors are simply more abundant on the afferent arteriole, so there is a net vasoconstriction of the afferent side more than the efferent side. Second big effect is stimulation of renin, and specifically release of renin from the juxtaglomerular granular cells, and subsequent activation of the full renin-angiotensin-aldosterone system. And that effect is mediated by the beta-1 adenoreceptors. And then third, the stimulation of additional sodium reabsorption takes place by activating tubular epithelial cells. This is kind of an interesting idea that a sympathetic nervous stimulation ending can quote-unquote activate an epithelial cell. But more specifically, what they're activating is the sodium-potassium ATPase that is expressed by those epithelial cells. And it does so through a signaling mechanism mediated by the alpha-1 adenoreceptor. The effect of activation of the sodium-potassium ATPase pump is subsequent additional reabsorption of sodium. And all three of these effects work together in concert with the big main goal of increasing blood pressure and trying to restore body systems. This slide is showing, once again, the three big effects of the sympathetic nervous system on local processes in the kidneys. They increase arterial resistance in the blood vessels, particularly afferent and efferent arterioles, with a more powerful vasoconstriction of the afferent side over the efferent side. Stimulates juxtaglomerular granular cells to release renin, and activates sodium-potassium ATPase of the tubule epithelial cells. And it does so through these receptors. So as you can see from the information about sympathetic stimulation, changing capillary hemodynamics via the sympathetics can alter things like filtration force and other Starling forces. And since those contribute to things like glomerular filtration rate and ultrafiltration pressure, we're going to circle back and start talking about what happens when you start manipulating capillary hemodynamics. So I'm going to briefly remind you of Starling forces. First Starling force is the hydrostatic pressure, denoted by the big P, in the glomerular capillary, so the GC. So hydrostatic pressure is kind of the pressure exerted by the fluid within the glomerular capillary that has a net kind of pushing outward effect. Now, hydrostatic pressure within Bowman's capsule or Bowman's space, that would be denoted by the big P and then either a BC or a BS. That's the kind of fluid pressure as exerted by Bowman's capsule or Bowman's space, kind of pushing back inward upon the capillary. Now, the next Starling force to take into consideration is the oncotic pressure, or oncotic, you can interchange the phrase colloid osmotic pressure. And the oncotic pressure is denoted by the symbol pi, and then oncotic pressure within the glomerular capillary is the GC. And oncotic pressure is the result of exertion of the kind of the pulling presence of proteins. So oncotic pressure tends to pull inward towards the capillary. Similarly, oncotic pressure as exerted by any proteins within Bowman's capsule would pull outward from the capillary. However, there shouldn't be any proteins in Bowman's capsule because proteins aren't supposed to filter across the glomerular filtration membrane. Therefore, under normal conditions, this should be essentially zero. Now here, we're going to begin to put some numbers to these values. So forces that favor filtration are the glomerular hydrostatic pressure, or the hydrostatic pressure in the glomerular capillary, and the oncotic pressure within Bowman's capsule. And as I mentioned immediately before this slide, that should be essentially zero. Now there are forces that oppose filtration. That would be the hydrostatic pressure within Bowman's capsule and the oncotic pressure within the glomerular capillary. So this is a graphic that depicts these numbers, and I just threw up some example numbers here to give you something to kind of solve for. So if you have your afferent arteriole, this is your glomerular capillary, Bowman's capsule, and the efferent arteriole. Forces that favor filtration are the glomerular hydrostatic pressure. Glomerular hydrostatic pressure tends to push outward. Okay. Forces that oppose filtration. The glomerular oncotic pressure tends to pull fluid in. And then there's a hydrostatic pressure within Bowman's capsule itself, which would kind of push against filtration. So in order for filtration to happen, the value for the forces favoring filtration has to be bigger than the value of the forces opposing filtration. So if you insert these kind of example numbers here, that net filtration pressure equals glomerular hydrostatic pressure minus the hydrostatic pressure in Bowman's capsule minus the oncotic pressure within the glomerular capillaries, you get a number. And if you insert 60 minus 18 minus 32, you get positive 10. If you get positive 10, then that means that net filtration is happening. If you had a negative number, that would indicate net filtration is not happening. So next we're going to start talking about changing the capillary hemodynamics and what impact that has on glomerular filtration rate. Three physical factors contribute to glomerular filtration rate, and they are 1, 2, 3 here. One, the hydraulic conductivity. This is basically how leaky is the fenestrated endothelium of the glomerular capillary. It's called the permeability or the porosity of it. Essentially, how leaky is it? The surface area for filtration. So you need to know how leaky is something and how much surface area do you have for stuff to leak through. Now, interestingly, the hydraulic conductivity times the surface area, those two, number 1 and number 2, multiplied together, give you something called the ultrafiltration coefficient, K sub F. This was mentioned earlier on in this lecture. So now you can go back and review that, and hopefully that will make sense. The third physical factor that contributes to glomerular filtration rate is the overall capillary ultrafiltration pressure, or PUF, ultrafiltration pressure. The product of these three factors, or the product of KF times PUF equals GFR. So these are the physical factors that contribute to glomerular filtration rate. And if you manipulate any of these three variables, then you can change GFR. So that leads to the question, what can change those three variables? Well, something that can easily be changed is the ultrafiltration pressure. Ultrafiltration pressure is calculated by the following equation, and you'll see this is something I'm trying to emphasize to you here. PUF equals the pressure in the glomerular capillary minus the pressure in Bowman's capsule minus the oncotic pressure in the glomerular capillary. So about 2 slides back, this is the same equation. So if you change any of these values, the hydrostatic pressure or the oncotic pressure, that can change the overall ultrafiltration pressure. If you change the overall ultrafiltration pressure, that can influence glomerular filtration rate. So an easy one of these that gets changed most frequently is the hydrostatic pressure in glomerular capillary. What can change that? Well, overall blood pressure in the renal artery, the resistance in the afferent arteriole, or the resistance in the efferent arteriole. You can also change the physical factors that contribute to glomerular filtration rate in terms of the ultrafiltration coefficient. Now recall a couple slides back, we defined the ultrafiltration coefficient as the hydraulic conductivity, or the kind of leakiness of the endothelium, times the surface area. So if you change either of those, you can change the ultrafiltration coefficient and therefore the glomerular filtration rate. So the question is, what can influence this variable? Well, what can change surface area? What can change permeability? What can change leakiness? Well, certainly if you damage the endothelium itself, that could change the leakiness. But another thing that is under good physiological regulation is the surface area. And this brings you back to kind of a question of what's the function of those glomerular mesangial cells within the glomerulus itself? Well, if you recall, those glomerular mesangial cells have contractile properties, which can literally help contract and change the surface area of the available endothelium that would otherwise be filtering things. So if you contract down those capillaries and make them smaller, then you're decreasing the surface area. If you decrease the surface area, then that can change ultrafiltration coefficient, which can influence GFR.